Good morning, evening, and afternoon, everyone, all around the world. My name is Payar. I'm the CEO and co-founder of Smart MCs. And today we are here with our second series of our webinar. And we are having a great host, Professor Majid Warkiani from UTS. He's one of also the founders of Smart MCs with a great focus on research focus on microfluidics, cell biology, and their translation into practice. So before I hand over to Majid, today's talk will be around 30 minutes, followed with a 10-minute technical questions and 10 minutes of career type questions for young entrepreneurs, innovators, and other people like the researchers active in the biotech sector. Without further ado, I will pass it on to Majid to give a quick introduction of himself, and very excited to hear his scientific talk.
Majid, thank you very much, Paya. Yeah, thanks, Paya, for your kind intro and arranging this series. So my name is Majid. I'm a professor here at UTS, and of course, one of the co-founders of Smart MC. I decided today give a glimpse of what I've been doing over the past decade around microfluidics and the journey I've gone through, and of course, the privilege that I had to be part of multiple startups in different capacity. So that's the snapshot that I put. I did my bachelor master in Iran. I did my PhD in Singapore and postdoc in US. I came to Australia around 10.5 years ago, started off at UNSW and then moved to UTS. And along the way, I had the opportunity to work with a couple of startups as listed here, which were companies that my supervisor spin out or was working as an advisor. And the last two that I will highlight today is the activity that we have done here back in UTS. During my PhD, I get exposed to the world of microfluidics, of course, and automation, where we were trying to essentially build a unique system for fully automated detection of pathogens in drinking waters, essentially shrinking multiple stage of filtration into a machine with a unique set of filters and a very dedicated PCR machine for ultra-fast detection of this type of pathogens. And of course, during that time, I learned a lot about MEMS, microfabrication, and microfluidics being part of this big project. And that's the machine that we built that time. It was an instrumentation with multiple inputs. My PhD was revolving around filters, isopore filters that we were utilizing microfabrication and MEMS to build them. To could utilize here, there was a stringent criteria to have a very good capture efficiency and release efficiency for PCRs. And later on, we built a miniaturized version of that for detection of foodborne pathogens through a lab on a chip system, as you can see in the top right corner. Later on, this technology evolved. When I left, I was still involved with some sort of consultancy with my former team. Part of these technologies commercialized through a company called StarArray. And more importantly, during the pandemic, because PCRs and ultra-fast detection of viruses like COVID-19 was so popular, this company managed to bring or to be one of the early companies to bring ultra-fast detection of viruses and pathogens to the market. And I believe there's still commercialization is happening through new generation of products, which you can see two of them here. When I started my postdoc, I started to be fascinated with the power of microfluidics in cell separation and liquid biopsy. I was working specifically a few years on isolation of the cancer cells, which I will allude more, and other type of circulating biomarkers for early cancer diagnostic back in US and Singapore. And along the way, over the past few years, we have generated several of these devices, which I will explain in details, using the concept of inertial microfluidics. And that time, of course, we had the opportunity to utilize a version of this machine and commercialize it through a startup back in Singapore called Clearbridge Biomedics. And that's the picture of that machine that developed that time. Of course, I've been scanning the field. And as you might appreciate, microfluidics influence a lot different fields. I put picture of a couple of these commercially available devices, each of which are influencing a big market, ranging from CAR T cells to NK cells for immunotherapy products, organ-on-a-chip systems for drug screening, platforms like the Berkeley Lights for single-cell assays and clone development, machines that they are developed nowadays with combination of AIs for next generation of flow cytometry, like the BDS-8, and of course, a lot of liquid biopsy tools for a variety of complications and malignancies such as cancer. My group specifically works on these three areas. So we heavily invested a lot on cell sorting and particle sorting, utilizing, of course, a lot of advanced microfabrication and microprinting to build those gadgets, and a little bit of works on around organ on chip, in which I put some examples for you today. So when it comes to biology and disease, we often works with cells, and cells are the building block of organs. And we know that when something's go haywire, something is a population of cells are not doing right. For example, in the case of cancers, normal epithelial cells started to proliferate abnormally in a very fast fashions, and they become cancer cells and spread in the body. Or in the case of cardiovascular disease or infection like malaria, parasite get into the blood cells and cause problems for the red blood cells. And often we need to find a tool to could isolate those malignant and disease cells. And these are three commonly used platforms in the laboratory if you have been working in a cell biology lab. Depending on the cell type of interest information that we have on that population or antigen status, we typically choose fluorescent approaches like FACS, or magnetic approaches like MAX, or density gradient, centrifugation, or even filtration systems. In the realm of microfluidics, of course, people have been trying to leverage on the same concept, but they have been trying to miniaturize to increase the sensitivity of the assays, the resolution of the separation, and build newer systems using multiple concepts that is illustrated through these schematics on the left-hand side. And today I will just focus on the inertial microfluidic side, but there are other opportunities and systems out there that people invested a lot of time on their theories and physics. And again, community has witnessed a surge of commercial device built using this concept, and they are already solving some important problems in different disciplines, including biology and medicine. When it comes to inertial microfluidics, there is a lot of work. So my group contributed a lot over the past 10 years of the physics and the background of how things operate in different type of inertia systems. We have published heavily on the physics, and of course, a lot of use case and application, which I will try to illustrate today. That's the most common shape of these channels that we have been using. Spiral shaped channels with typically single inlets and two outlets, where we can play with the physics and of course geometry of the channels, especially the cross sections, to build useful gadgets for particle and cell separation. And of course, we can build these things through traditional CNC cutting and PDMS casting, through the 3D printing, and more recently we can do even injection molding of them for larger scales manufacturing. One of the key areas that I mentioned that we have had a lot of interest and the field has been growing is the field of liquid biopsy, where cancer biologists and of course clinicians are interested to find some useful information about the cancer as early as possible. So we know that by now cancer cells started to shed information into the blood circulation or other bodily fluids through the format of normal cancer cells or the clusters, or even breakdown of them, such as cancer DNA or called ctDNA, and a lot of other soluble factors and proteins or microRNAs in different shapes and format. And on top of that, the field of exosomes, which I will highlight that more in details, is becoming so popular on set of liquid biopsy. The promise of liquid biopsy compared to the traditional biopsy is that we can utilize these systems in a very dynamic fashions. We don't need to wait a few months. We can do collections on a weekly basis and get real-time information about the cancer development. And of course, we can utilize them to personalize the treatment. And more importantly, liquid biopsies are way more less invasive compared to the traditional biopsies. And again, there is a lot of debates, but in my opinion, liquid biopsy came to complement traditional biopsy, and nowadays the modern cancer management is around that, and we are trying to see how we can utilize them in the clinical setting. When it comes to the actual cancer cells and the way the cancer is metastasized, typically cancer cells find their way into the circulation in a format of individual or clusters, and they can transfer through the lymph node or circulatory systems and find a way to the secondary organ. And that's the way the cancer cells spread in the body. And of course, over the past decades, a lot of systems, a lot of microfluidics and non-microfluidic system has been developed to isolate these rare cells from circulation, which you can read a lot of review articles about them, like the one that I mentioned here. My group, of course, heavily invested on the inertia side, and over the I would say decades, we have built different versions of these platforms, different channel geometries, each of which designed to enhance the separation efficiency, working with the lice blood or food blood. And of course, the most critical aspect is that how this type of platform performs in the clinical setting. And again, we have been fortunate to collaborate with eminent scientists across the globe and validate this system. So we have tested these devices across multiple solid tumors, breast cancer, lung cancer, colorectal, glioblastoma, and many others. And of course, in the clinical side of the stories, we have been trying to see how we can find more useful information compared to the traditional biopsies using this system, which I will illustrate some examples. Not only with the blood, these systems, given their capacity to handle larger volumes, we have tested them for other biological fluids, such as urine, that you can isolate the cancer cells for the case of prostate and bladder cancers, or even the seminal fluid, which is a very, very viscous type of fluids, which is hard to handle, but we have showcased recently that the possibility of isolating tumor cells from the seminal fluid for the later stage prostate cancers. It's few years that the field has been very fascinated with the context of CTC clusters and the way that they form or shed into the circulation. There has been some seminal works with Nicholas Aceto and Mehmet Toners discussing the tumorigenicity of the clusters and the fact that compared to the individual CTC clusters can be much more dangerous in terms of metastasis. And of course, a lot of theories, as you can see by Klaus, a recent report by Klaus, how these clusters are forms. Either they are homogeneous or heterogeneous in terms of carrying platelets or the blood cells. Do they form in circulation or do they detach as a group from the tumor itself? These are type of theories that we are trying to address nowadays. These are some recent data that we published with a group of scientists back in the University of Queensland on the role of macrophages and the way that they escort the cancer cells and form a very dangerous format of clusters when it comes to the lung cancer metastasis. And what we are trying to do is that this type of homo and heterotypic clusters, we try to see how their count and presence fluctuate across different type of patient at different time points, and whether we can build the diagnostics around the presence and their analysis using advanced genomics tools and multi-omics tools that we are trying to put together. And surprisingly, when we started to do IHC and high resolution tissue, the same format of clusters that you can see here that we see in circulation, we do see in the tissue also in terms of communication that they do, suggesting the idea of the fact that this type of clusters can be detached as a group from the primary tumors and keep the shape format when it comes to the circulation. Of course, when we want to analyze this type of complicated structures, we need proper tools. And again, the field of single cell analysis has been a flourishing area of research. Several companies and products like the 10x Genomics machine or BD Rhapsody came to the market, each of which with the promise of enhancing the field of single cell analysis, but through systematic analysis, we realized that while these platforms are very good for the case of high throughput single cell analysis, when you have a load of them, when it comes to the rare events like CTC and CTC clusters, it's pretty hard to utilize this system. And that's why, of course, Payar himself reviewed this field. More recently, you can refer to these review articles talking about the history of single cell genomics and how each of this modality helped to understand the biology of different diseases, including cancers. But what we were trying to do ourselves is to harness the power of microfluidics, especially static droplet microfluidics, like the one that you see here, where an easy to operate device like this PDMS lab could facilitate essentially injection of a known number of cancer cells post isolation, and of course their assessment using a variety of biological tools such as immunostaining, or more recently, we showcased the possibility of doing metabolomics screening like that. As you can see, these devices are very easy to operate. We are leveraging on the Gecko features of the PDMS in contact with a solid surface like glass. And after the formation of these tiny pico-size and nano-size droplet, we can snap freeze them on a dry ice, and essentially giving the hydrophobicity of these materials, we can lift off and have direct access to those individual droplets, where you can analyze captured cells or clusters within them. As I said, one of the critical challenges in liquid biopsy has been the idea of how cellular staining, such as cytocroting and HER2 staining, or even EpCAM, is indicative of a cell being cancer cell, and can we add another layer of modality to confirm if a captured cells are cancer cells or not. And that's why we leverage on the idea of glucose consumptions and showcase the possibility of utilizing a system like this to monitor in real time the amount of glucose that the cancer cells consume compared to the immune cells. And because we know that glucose consumption and metabolomics is a hallmark of cancers, we can essentially, in a label-free fashion, distinguish between those pockets that they hold cancer cells from those pockets that they don't have cancer cells. And of course, this has been rigorously tested across various cell lines and different model of the breast cancers, syngeneic, modic, transgenic, and non-metastatic ones. And we could see very clear distinctions in terms of the metabolomics behavior, in which I recommend you read the papers in details if you like. Beside the CTC and CTC clusters, the field of small extracellular vesicles or exosomes is pretty hot topics. People are actively looking to utilize them either as a diagnostic tool or as a therapeutic. And my group has been trying to come up with some innovative idea of enriching them for the diagnostic purposes. And if you follow the fields, you know that this field has attracted lots of attentions. 2 Nobel Prize has been given for the case of exosome 1 about their regulate and vesicle trafficking in 2013, and more recently about the idea of microRNAs, how microRNAs can essentially cause translation of the proteins, which was given to the Gary and Victor last years. And again, the field of microRNA assessment, especially exosomal microRNA for the diagnostic purposes, is very, very hot topics. This is an example of a recently developed test called ExoDex for the prostate cancers, where the company come up with a panel of biomarkers extracted from exosomes enriched from urine samples of the prostate cancer for early detection and monitoring of the prostate cancers. And then the same modality has been applied for other type of solid tumors. We are doing a work on this for mesothelioma and lung cancers in my group to come up with a unique set of exosomal microRNA for early detection of the lung cancers. But one of the biggest challenges in the field of EV and EV research, similar to the cell side of the story, is the heterogeneity, where we know that these few years that we appreciate that when the tumors is assessed at different locations, different type of zones, they can express different genetic profiles, and that special heterogeneity that we see within the tumors is present in the tumor drive materials, including CTCs and exosomes, and that has been made the field of diagnostics pretty challenging, and of course people are looking for tools to address it. For example, one of the big challenges past few years ago has been the direct flow cytometry assessment of them, because these are very tiny nano-sized particles. It's very hard to use traditional flow cytometries to assess their surface markers, and that's why we built a unique coating using metal organic frameworks 3 years ago to enhance the capture efficiency of the EVs on top of the beads, polystyrene beads, to boost the sensitivity of their detection using flow cytometry. Or more recently, we analyze and assess the difference between different type of antibodies and antibodies as a new small molecules for detection of EVs. And we are collaborating now with a company in China to utilize a direct assessment of them using a new generation of nanoflow cytometers for the field of cancer and cancer research using EVs. When it comes to the subpopulations, for example, for the case of breast cancers, people often interested on the proteins such as HER2 or EpCAMs. And when you want to look for tools to could isolate the subpopulation of EVs that express potential biomarkers, it is pretty challenging. And that's why we build an inertial microfluidic systems combining with the immunoaffinity captures to could address it. So the way that we have done it is that we come up with beads of different size, 3, 10, 20 microns, decorated their surface with the capture antibodies and incubated with those samples, such as the plasma. And then using a device like these, we can easily, based on the size, separate beads of different size that they carry those type of subpopulation of EVs. And in a very low-cost fashion, we can enrich EVs that are positive, for example, for PD-L1, EVs that are positive for HER2, and the EpCAM for downstream assessments. Of course, we have done a lot of characterization to showcase these methods is sensitive compared to the traditional cytometry, subpopulation sorting, or magnetic beads. And of course, in terms of cost efficiency, our method is proven to be much cheaper compared to the traditional methods. And of course, a lot of fundamental works around the roles. When you get the subpopulation out, how they cause changes in cancer on the context of epithelial to mesenchymal transition, we have an active area of research where we're trying to see how EV influence EMT and EMT related markets. Of course, the microfluidic usage is not just limited to the cancers. Smart MC has been trying to leverage on the power of tissue engineering, biomaterials, and microfluidics to address some challenges. And I've been lucky to collaborate with the team on utilization of the microfluidics for these purposes. One of those things, of course, is large-scale manufacturing of adherent cells in different industry, ranging from vaccine to cultured meat to a stem cell itself. And we know that when you want to scale up, the traditional approaches of 2D culture are not enough. We need to use 3D approaches. One of those 3D approaches is adherent cultures inside the bioreactors. And when we scan the field over the past decades, we have seen that the field of microcarriers, in which has been an active area of research, has been stagnated around products that are not really customizable nor designed to handle different cell types. And that's what we wanted to explore, the idea of customizability, to build cell type specific products. Depending on the origin tissue, we can fine tune the properties of these carriers to could utilize them in a very efficient fashion within bioreactors. And there is a lot of knowledge now out there discussing the idea of the fact that the substrate stiffness and property influence the quality of the cells and their byproducts, including exosomes. And scientists are paying more attention on utilization of proper materials for different applications, particularly around usage of them as a therapeutics, such as mesenchymal stem cells and iPSCs. And of course, again, microfluidics has been the workhorse here for us to leverage on it and scale up in a sense that we could build these customizable platforms. And what we're trying to promote here is that the time to do 2D cultures and use a lot of plastics and waste a lot of materials is over. Now, in a very low-cost fashion, in much smaller footprints, we can really do enhance cellular production, especially for adherent cells. This is an active area of research, and of course, given the burst of cellular therapies, especially MSCs, over the past 24 months, we do see a lot of interest for scientists to transit from the 2D to 3D culture, and that's what we try to offer here. And at the same time, we have tested, of course, this type of products, compare them with the existing solution in the markets in terms of the performance, which you can liaise with the Smart MC teams. These are some of the recent data which are under publication for different type of adherent cells, such as MSCs and the fibroblastic cells, where we have showcased that matrix stiffness can influence the production, and we have fine-tuned the best substrate that can be utilized for production of fibroblastic cells in terms of synthetic meat and the science of alternative proteins. So that was the second example where microfluidics can be utilized, especially droplet microfluidics. But that's not the area that we have been doing. We have been exploring over the past 5 years the role of microfluidics and what it can do or help in context of fertility. Again, infertility has been on the rise, and the male side of the story has been ignored. And there's a lot of innovation on the female side, but we feel that we can fill the gap with microfluidics. And again, when you look at the spectrum of technologies that can be utilized or developed in infertility field, there's a lot of advancement and development, in which I will give you two examples. One of those things is the field of sperm isolation and analysis, where we have seen less improvement, and still the gold standard is traditional centrifugation and swim up, where there's a lot of evidence nowadays showing that these type of methods can cause DNA damage, and DNA damage directly translate to lower implantation rate, poor quality, and higher miscarriage rates. And that's why we have been trying to look at the mother of nature, explore the possibility of utilizing microfluidics to mimic how human body essentially do sperm selections. From the time of the sperm production to the deposition and essentially transport across the uterus junctions toward the ovary and ovary gland and essentially eggs, there is multiple scenario of selection happening. It's called tegmotaxis, rehotaxis, thermotaxis, and chemotaxis. And of course, each of them play an important role in this space. The one that we have been utilizing to mimic in microfluidics is called tegmotaxis, which is a boundary following behavior of sperms across different microstructures, in which for different species has been explored. And what we did here, essentially, we tried to mimic those microstructures using the 3D printing technology within a microfluidic cartridge, like the one that you see here. We know that the sperms, when it reach a confined environment like this, they try to transverse from the left to right, and that transverse is essentially a function of the DNA integrity. And of course, we can add second layer of filtration in these platforms, like the one that you can see here. At the middles, we can put beads coated with Annex NV to get the apoptotic one. But the idea here has been how we can build a simple cartridge like this, where you can deposit a raw semen, wait 15 to 20 minutes, and collect high quality grade A essentially sperms from the centers. This type of work is already published. You can have a look. But what we have been trying to essentially achieve here is an easy to use, or a format that can be accessible to our embryologists, and is not really hard to operate, as easy as centrifugations. And that's the journey we have gone through, from early prototyping using 3D printings to the soft lithography back and forth. And of course, more recently, we have built an open version of this platform using the open microfluidics scenario and injection moldings to build these gadgets in devices that they are gamete friendly and FDA approved, in which another startups in Sydney called Neogenics is planning to commercialize them as a Class 1 medical devices for andrology lab across the globe. And the benefits that, of course, we bring to the clinics is this. You can see that traditionally, there are multiple steps involved for semen preparation and sperm isolation, and a lot of time. And of course, a lot of variability across labs. So what we're trying to achieve here is shortening time, enhancing the quality of the sperm, because it's a biomedical approach, and more importantly, a way to standardize the entire process of sperm selection across different labs in different countries. And we are hoping that by adaptation of these technologies, we could see improvement in statistics of the IVF across the globe. Of course, a lot of clinical data to back the hypothesis and theory, but one thing that you need to pay attention is this important factor called DFI, where by utilizing microfluidics, we can drop the DNA fragmentation index down to 80%, which is pretty significant in the clinical setting, yet to be determined in a long-term clinical trials. And not that, we have showcased the possibility of essentially utilizing this type of systems for cryobanks, where sperm donation is happening, but raw semen cryopreservation is the norm. What we're trying to propose here, if we could enrich the sperms and then cryopreserve it for longer times, you do get a better quality result post-time for clinical settings. which is an active area of research here. At the same times, what we have been trying to help a group of patients that they do not have actually semen in their samples, and they need to go through invasive surgery and biopsy called microsurgical testicular sperm extraction, where often the tissue should be explored and assessed under the microscopes by a trained embryologist for a few hours to could find a very rare amount of sperms if presents there. So what we did recently is that we utilized AI to train a program to could help embryologists to find the sperms in these complicated samples much faster. That's an early version of the system showcasing that while human eye cannot really see the sperms or the presence of the sperms in this complicated type of tissue, AIs in a very fast fashion can detect and alert the embryologist and help to shorten the time for searching surgical samples to find suitable candidate for the ICSI procedure, which was recently also published and is under commercialization by Neogenics. And what you love to look here on the age of AI is the amount of time that you're shortening with AI, and more importantly, the level of accuracy and precision that you get with this type of platforms. So far, how much time I do have left, Paya?
You can go for another 10 to 15 minutes. That's okay.
Okay, that's good. So the last part that I would love to show you is around another field of microfluidics. It's called organ-on-a-chips. In more specific fashion, we have been trying to build tumor-on-a-chip models. And you might appreciate that after decades of the research on the 2D culture systems, and of course, animal models, we started to really appreciate more that how incompetent are these models for advanced drug developments. Also, there's a lot of discoveries, but we know that 2D cultures and animal models are not good resembles. And of course, it's a decades or more people are trying to utilize the power of stem cell biology and organoids to do that. And each of these models, which are the workhorse in the pharmaceutical industry for drug candidate development and screening, have their own drawbacks. And that's why three important fields emerged, and now they are combining the power together to enhance the way that we do drug screening. Organoids has been an active area, as I said. 3D bioprinting is another area which is flourishing, and a lot of developments happening here, especially around the biomaterials and the field of organ-on-a-chips. And the synergistic effect of them, in my opinion, in the next 10 years will be the winner for advanced models for drug screening. And a testimony to that is hundreds of companies, the one that some of them I listed here, is that they are emerge, each of them are trying to offer a new solution from pharma and essentially fundamental biological research using these models. This is a snapshot of, again, some of these microfluidic enabled products out there, the cartridge developed by Mimetas, the one that developed with Alveolix, Tissue Use, and Emulate Bio. All of them, they are harnessing the power of automation, AI, and more importantly, microfluidics to build system for drug screening. So I've been lucky to know one of these companies called Biotechs. We have been collaborating over the past few years to build more knowledge around their unique systems, the 3D culture models developed by Roger Kam back in MIT, and my friend Amir Reza, and heavily tested by some good cancer biologists like Jean-Paul Thierry. And of course, there's a lot of knowledge around them nowadays in the market, a lot of protocols to use, but still we're trying to see how we can build more useful information utilizing these systems, how we can test new generation of biomaterials, synthetic biomaterials for the tumor and organoid research using these models in which Smart MC is trying to influence and produce data around that too. As you can see, very, very sophisticated questions such as the process of EMT, the way that cancer cells lose their property, epithelial property, and become mesenchymal and spread in the body can be captured, or even a complicated scenario like vasculogenesis or formation of lumens can be monitored using this system, in which a few years ago was not possible using traditional approach. I put 3 case examples for you, how we utilize them to give you a glimpse of what can be done. and where the field is going. One was a collaboration with a company called Novocure, which is commercializing and got FDA approval for a new generation of treatments using electric field. This company has been researching the idea of how we can use external fields to deliver low intensity in a range of 1 to 3 volt per centimeter intermediate frequency to kill the cancer cells. And of course, if you go to their website, you can see they have been a very successful company, bringing several FDA approved products into the market, and they have so many clinical trials going on. The way that this technology works is that they have an array of electrodes depending on the type of tumor. Either is for the glioblastoma, they put it on the head like this, or for the case of lung cancers, they put it as a stitch. And you have a wearable gadget where it apply low intensity electric field. And the biology behind it is that we know that when cells go under the process of cell division called mitosis, during the envelope formation, spindle formation, an internal electric field is happening. And when you put an external electric field around it, you're essentially forcing the cells to go toward apoptosis signals. This is a normal. Panel A is a normal cell division, but panel B and C are abnormal cell division. And that's a movie of how mitosis actually is happening in action. So in short, this company claimed that by applying electric field at certain frequency, we can disrupt the formation or cell division, which is the hallmark of the cancers. We know that cancer cells divide much faster than the normal cells at different frequency. If we attack them at that frequency, we can disrupt their cell division while we are not affecting the division of the normal cells. That's the way that they used to do and is still doing 2D models, culturing cancer cells in a spheroid or an individual format, applying electric field. That's what we did with the power of microfluidics. We essentially built a gadget with integrated electrode to model how things works at a much smaller scales. And of course, we have tested this for 2 years. Initially, we were not getting much of results, but eventually we realized that what they're claiming is correct. There is a sweet spot of frequencies where if you use a normal cells like a Huvik or a cancer cells, you can apply these signals to give apoptosis to the cancer cells and stop their proliferation. Again, this is an active area of research. The company published excessively around this idea, but what we did, we wanted to see how microfluidics essentially can help them to better understand their theory, in which ended up being a joint publication with that company. On the same note, around cancers, another active area is cancer and cancer microenvironment. We know that the tumor microenvironment is a complicated tissue full of cells with different properties, each of which can have anti or pro-inflammatory properties. One of those cells that has been the center of attention is cancer-associated fibroblasts, in which people have been trying to target them as a new modality to enhance the therapeutic efficacy of the traditional chemotherapy or modern immunotherapy. So what we did a few years ago, we have analyzed TNBC, triple negative breast cancers microenvironment, and we wanted to explore the possibility of utilizing an anti-fibrotic agent such as pirfenidone as a complementary tools to modulate tumor microenvironment and enhance the receptivity of the cancer cells toward traditional chemotherapies. Of course, again, we utilize microfluidics and we build models to could assess their roles, how essentially CAFs induce invasion and immune suppression. We can clearly see in these models compared to the traditional 2Ds, and how they affect the cytokine, pro-inflammatory and anti-inflammatory cytokine productions. Which of those cytokines essentially have more immunosuppressive roles? And more recently, I realized that a follow-up of our work is done in China, where peoples are utilize these to treat breast cancer models, triple negative breast cancer models, looking at the potential signaling pathway of TGF-beta SMAP. And as you can see, the data that we generated has been confirmed and perfectly validated in this new publication, showcasing the possibility of combining an anti-fibrotic agent with chemotherapy to boost the quality of the cancer management using the traditional and immuno-oncological drug such as PD-1, PD-L1 platforms. The last examples that I want to show you is the idea of organotropism and how we can utilize microfluidics to look at this. So we know that by now, after many years assessing different type of tumors, for example, for the case of breast cancers, we know that typically breast cancers love to metastasize to the brain or to the liver or bone, but we still don't know why and how fast that is happening, and in which stages we can detect or prevent it, given the knowledge that we have. So what we have been trying to assess more recently is the role of exosomes and how exosomes essentially educate or facilitate organotropism, the way that the cancer cells decide to metastasize from one organ to another organ. So what we have done here is that we try to utilize the power of microfluidics and organoid, combining them together to understand really how exosomes drive, for example, from an organ such as brain, can educate cancer cells and generate a memory inside them to could penetrate or migrate toward a particular organ. As you can see in this experiment, we cultured a large body of organoids, brain organoids, in collaboration with Anna from University of Sydney, and isolated a large volume of this brain-derived organoid supernatant, extracted the EVs, and then educate the cancer cells using these EVs, and looking at how brain-derived organoids can influence the migratory behavior of the cancer cells in these models. Using this type of microfluidics, as I showed you before, we can systematically assess them. For example, for this case, we have utilized MDA MB231 cells to build a normal and educated version, and looking at the transmigration behavior of them when they are co-cultured with the brain organoids again. And we confirm that those cells that they are educated with the cancer drive, with the brain drive organoids have higher tendency to move toward the normal brains compared to the uneducated one, confirming the theory of exosomes play a key role in the organotropism and how cancer cells spread or choose a particular organs to metastasize. Of course, the idea around translation of these technologies after doing all these fundamental works is to combine liquid biopsy and these 3D models to build a better tools for personalized cancer management. This is an area that we are looking into it to see how we can bring some sort of test and products into the market. Some more recent works done in Boston by Dr. Arif, showcasing, for example, the same models can be utilized to look at the efficacy of the immunotropic candidates, such as the PD-L1 models. As you might know, immunotherapy treatments are pretty expensive, and finding the right candidate who could benefit is very, very important because before administering such expensive drugs. And this type of microfluidic models are used now as an ex vivo type system to do drug screening prior to prescribing such expensive treatment for the patient and looking at their efficacy. And that's the future, the way that microfluidics and biology are helping to build new modalities for better management of the cancers toward personalization. So with that, I hope I'll give you enough examples how microfluidics can be influential or can be translated into the market to our useful products. These are all testimony of the hardworking student over the past decade doing all these works, a lot of collaboration, a lot of funding from different agencies, as you can see here. I would love to acknowledge their contribution and take questions if you have any. Thank you very much.
Thank you for that excellent talk, and I think it was definitely full of different disciplines along with many different topics and applications, which makes it super exciting. So we already have a couple questions from the audience. So I think Abanub is asking about your spiral microfluidics. So first of all, we thank your presentation for the great works, and he has a question regarding spiral inertial microfluidic devices. He's asking if there are any limits on the cell particle size that can be separated using that, and also if it can handle viscous liquids.
That's a good question. So yes, inertial microfluidics is typically good with the micron size cells. When you go to the nano size is because of the Brownian motion effect. you cannot really do efficient sorting. So when you're dealing with a micron-sized particles, is pretty good. When it comes to separation, you need to really have a few micron differences in size to could separate them. Let's say if a cell size that is 3 micron from 6 micron, you can use it, but if it's 3 from 4 micron, it's pretty difficult to do with inertia. But other systems such as DLD, deterministic lateral displacer system, are more suitable for that. In terms of handling viscosity, yes, you can run both Newtonian and non-Newtonian fluids inside. We have run blood or even seminal fluids with them. Of course, the cellular crowding can influence the separation efficiency. You need to really work on the range of 1 to 2% when it comes to the cellular concentration. But other than that, it's pretty high throughput systems for a variety of application.
One question there about non-Newtonian fluids. How do you see them differently? Like how particles behave differently in those type of fluids? Like I assume when you go on higher throughput with non-Newtonian fluids, there might be different behavior of the cell
Yes, of course.
separation.
Yeah, that's right. So a new generation of forces, non-Newtonian forces, is kicking, which act in a opposite direction of the inertial forces. Of course, if you fine tune it, you can leverage on it to enhance the resolution, but most of the times it disturbs the way that we do the self-focusing and essentially not helping much, put it this way, when it comes to the separation. And at the same time, preparing non-Newtonian fluids itself can cause some cross-contamination for diagnostic assays. So I haven't seen really a non-Newtonian approach come to inertial microfluidics and be translated. It just has been some basic biological and physics around it.
Yeah, that's cool. Excellent. Thanks. We've got a question from Yusuf. So, what methods are available for calculating the concentration of exosomes in suspension sample? In most papers, nanoparticle tracking analysis or NTA have been suggested, but I would like to know if methods like dynamic light scattering, DLS, or flow cytometry have sufficient accuracy for this purpose.
So DLS, to some degree, yes. Flow cytometry, absolutely not. It's not a good method to do concentration analysis. At this stage, an NTA approaches is the gold standard still, which works on the basics of Brownian motion assessment. But combination of DLS and NTA with some modern image analysis, AI-based systems that they are trained to do nanoparticle count analysis is coming to the market. I believe that's going to be the most accurate one, which we will see in the future.
Excellent. And on the separation of sperm cells, Yusuf is asking if when you're sorting your sperm essentially by using a microfluidic device, the sperm cell sample of each individual may vary, especially in terms of motility and number of cells in the raw sample. Is it necessary to design a customized device based on the sample specification?
Not really. I mean, it's going to be a hard task, but the reality is that there is a global swimming pattern across all the sperms. This is their nature and they can, but again, of course, that level of heterogeneity in the individual you do see. But given the fact that typically majority of the patient have a lot of Siemens in the samples, you do see a good yield when it comes to the separation efficiency. Again, I'm not sure if personalization of the cartridges is a good approach to look at it, given the associated costs in entire IVF cycles. So we feel that a generic device like this that can handle a variety of samples like that generate enough yield to be used in the clinics for those type of fertility treatments.
Great, thanks for that. I had actually one question on the cancer side. So you've done a lot of work on the circulating tumor cells and some more on the exosomes. And then you've done some other works on organoids. Where do you see them interconnected? So are there different pieces of a diagnosis, such prognosis aspects in clinical settings, or do they just play different roles for different types of patients?
I believe, again, it will come to the use cases where you want to have such a tools accessible in the clinics or hospitals, fully validated, FDA approved. But you want to depends on the question that the oncologist has. Sometimes they really like to use them as a method to look at the minimum residual disease if the patient has gone the treatment and if any residual disease remain. So you use liquid biopsy, either ctDNA or CTC for that, or you want to get a definitive answer around a doubt that you have. For example, the biopsy for the tissue, for example, HER2 came negative, but the CTC HER2 status is positive. And now there is a debate that which kind of treatment we give. If the liquid biopsy positive, tissue negatives, how patient can benefit. That's where we can utilize liquid biopsy as a second confirmatory for selection of the treatment for the cancer patient. On the organoid side, it's much more new, but the example that I give you and many company works is around how we can essentially choose the right drug for the right patient before administrating or try and error, especially for expensive candidates such as CAR T cells or immunotherapy drugs. We want to see which group of patients are benefiting, and doing a biopsy or doing a liquid biopsy to good, looking at those predictive biomarkers around them can save a lot of time and energy for the patient and the families. That's where I see organoid and tumor on a chip models could be useful. And of course, when they become more mature and more cheap to run, combination of them going to be the best solution, liquid biopsy and organoid on a chip.
Excellent, thank you. So just before we hop off, I thought we asked questions on side of the carrier and how your research went. I assume you had good exposure to 3 different countries: US, Singapore, and Australia in terms of like the research ecosystem, funding sources, and all sort of different types of activities that goes on really in the academic world. Wanted to first of all hear a little bit about the differences along each country that we have seen, and also how the time has changed. So I guess over the past 10 years, has the fundings been reduced overall? Are there more pressure, or is it harder now for new researchers to get in academia for now, or is it vice versa? Just wanted to get a bit of insight on that.
Yeah, so that's a good question, Pyar. So look, academic jobs has been historically always difficult to get, or when you get it, difficult to survive, because of the fact that you need to often search for grants and research money to do good science. And different countries have different agenda. I mean, the budget of research in some countries like Singapore or Korea is very different with the one that you do see in Middle East or even in Australia, New Zealand, or compared to US. Typically, US has way more funding agencies in terms of the source of the fund, but of course competition is fierce because there are so many universities and academics out there. Australia, Canada have almost the same pattern. There is a relatively good amount of research money compared to the GDP available. If you ask us, we always say it's not enough. If you ask government, he said more than enough. So it depends whom you ask. Again, the success rate, I would say, across the globe for achieving such a grant is almost the same, 10 to 20%. When I ask people in China or in Korea, they need to fight. And typically success for getting such a grant is like that. Which country is better for research? Of course, countries where translation is appreciated more or is really knowledge driven are better, because the path to commercialization is much faster and there's a lot of resources around that. But when it comes to the balance of life and work, you want to choose where to live. Of course, Australia can be a good example of having benefit of both worlds. In my opinion, a young person who graduates should explore, like me, multiple places. Do not bind yourself to one places, because life is too short to do that. So, but at the end of the day, when you want to settle based on those experiences, you choose the right place. If you started in Australia, of course, it's good to have exposure of Asia and North America. Down the road, if you started North America, vice versa. So I highly recommend that you spend a few years of searching around and getting exposure of different countries to build a career for yourself down the road.
Would you say there's still the same amount of opportunity almost to get into academia, or is it getting less or harder?
If you're really keen and very, very determined to do that, the path is very clear in different countries. There is, of course, opportunities out there and new universities are coming. You should not expect to often start off a very high quality university. You can start from a junior or a relatively young university, build your career around that and come back to the top universities. That could be a good approach for those, but I generally recommend that if you have done a PhD, get an industry exposure, then decide come back to industry or stay in industry rather than staying for whole life in academia or in industry, both of them.
Great, great. Okay. And the question Lynn asked, so what recommendation would you give to young scientists who's trying to explore a new field? How do you build that credibility and connections to step into it with a very limited resources?
Collaboration. Again, you get some experiences. The best way to get into something new is academic collaboration, voluntary works, a lot of readings and engagement with the community that you would love to get in, and never scare of that. I myself, 5 years ago, had zero knowledge around IVF and sperm and everything. But again, we managed to collaborate with some good peoples in Australia and globally and get to this field and start to find the gaps and identify some solutions. I believe there is always options to get in, but the best way is academic collaboration, which most people are open into it if you have something to offer from your skill set. So that's the best way moving forward.
Yeah, great. And a general question, when do you think is the right time in terms of the research career to translate something? I guess it can be different for different applications and technologies, but is it better to kind of think about translational stuff early on in the career or later on towards more senior life
Honestly,
time?
it depends of the environment that you grow up and you do your PhD, especially. Some people's ended up in their bachelor even be in a good group and spin out something out of their bachelor works. And some people's need to do several postdocs until they find the right environment. That environment that you choose to be in dictates that for sure. Other than that is the personal perseverance and ambitions that you have. But certainly there is a lot of problems to be solved, and they need to be solved by people who have PhD. 99% of community that we live with, they don't have the knowledge of doing this stuff. So we do have the knowledge, skill sets. All you need to do is to identify the right problem, build a proper team or a group of people together, attend some of these accelerator type programs to get exposure around entrepreneurship, and build upon that. And you need to be sure that the first attempts or second attempts is not going to be 100% successful. It's going to give you experience. But if you be successful in the first attempt, so sum up. So that's a great thing. But that has been happening for myself. So I've been seeing a lot of startup over the past 10 years. We had a lot of failures ourselves. And now after 10 years, we know that what is right and what is wrong to do in terms of entrepreneurship, and hopefully it's going to get better and better in the next 10 years.
Certainly, I think that's definitely a very exciting journey of seeing also many different like disciplines that you have worked on and different I guess applications of microfluidics particularly that you have been focused on. So thanks a lot for today's presentation. I think that was a really fantastic eye-opening for a lot of people for sure. We'll definitely put this recording online on YouTube as well so people can watch it later. So if there's no other last minute questions, I guess we are right on time on the clock. So is there anything else, Majid, you wanted to say or share
No,
with
thank you
people?
very much. No, that's great. Thank you very much. I hope this series goes very well and more people come. Looking forward watching those future presentations, hopefully from eminent scientists across the Australian globe. And thanks for giving me the opportunity to showcase some of our work.
Excellent. And thank you so much for putting all the time. And thanks everyone for watching, and we'll see you
Cheers,
in the
guys.
future.
Bye-bye.